Today, I would like to talk about why it's important to always consider metabolites when analysing for trace organic residues. That is, things such as pesticide residues, sports anti-doping, or residues of organic contaminants in the environment. I'll start by giving a very broad definition of what metabolites are in this context. I'll then give some reasons as to why they should be included in the measurement of a particular residue, but just as importantly, where particular metabolites should be excluded. This decision as to which metabolites to include when measuring a residue is absolutely critical. Finally, I'll explain some different ways to measure and sum the chosen metabolites. A metabolite, in its broadest terms, is any byproduct of the biological action of a chemical. If you think of the types of chemicals we're talking about here, pesticides, veterinary drugs, pharmaceuticals, almost by definition, they all have a biological action of some kind. When a herbicide attacks a weed, metabolites will form. When an antibiotic attacks bacteria, metabolites will form. Metabolites are also a mechanism of organisms clearing chemicals from their system. In the case of animals, for example, the chemical might be converted to something more water-soluble so that it can be excreted in the urine, or it might be converted to something more fat-soluble or bound to a protein so that it can be deposited long-term in the animal's fat or internal organs. It is important to remember that we're not just talking about plants and animals, though. One of the most important sources of metabolite formation are microorganisms, things like soil bacteria. When we talk about environmental breakdown products or degradation products of a chemical, more often than not, we're talking about metabolites. So it is unlikely that any residues of an organic chemical in the environment or food will exist purely as the parent molecule. Most will include a mix of metabolites formed by different routes. These metabolites may or may not be significant. As a simple example, the insecticide DDT is incredibly persistent in the environment. This persistence is the reason that it's banned in most parts of the world. But it's not actually the parent molecule itself that persists. It is mainly a mix of two metabolites formed under two different conditions. In the absence of oxygen, so for example in sediments, DDT will metabolize in microbes to DDD, also called TDE, just to confuse people with as many three-letter abbreviations as possible. As here, you can see, if you look closely, that one chlorine has been replaced with a hydrogen. But more commonly, in the presence of oxygen, it will metabolize by a different route to form DDE. This is what happens in animals, with DDE deposited in the fat, or in microorganisms in the presence of air and sunlight. DDE bioaccumulates in the food chain. Despite DDT having been banned now for over 40 years, you can still find DDE in animal fats from almost anywhere in the world, and still with elevated levels in food originating from where DDT was used more extensively, such as New Zealand. DDD and DDE are two relatively simple examples from the metabolism of DDT, which can itself be quite complex. But metabolism within animals is generally more complex still. Don't worry, I'm not going to test you on this diagram. It's just to illustrate this complexity with naproxen, an anti-inflammatory drug. In this case, it's in rats. Every animal species may be different. But the common factor I want to draw out is that metabolism is in two stages. Phase 1 metabolism is the byproduct from the biological effect. So in this case, naproxen is converted to DMN. Phase 2 metabolism is converting the DMN to a more water-soluble molecule so that it can be cleaned from the animal's system. This is sometimes called conjugation, the molecule is bound onto a big water-soluble group like a glucuronide or a sulfide, so that it can be excreted in the urine. Metabolism also occurs in plants. This is particularly relevant for pesticide residues. Pretty much all pesticides will form an array of different metabolites, and this will be different depending upon the plant. So for example, pyrimecarb is an insecticide. In most plants, its main metabolites are the top two compounds here that are circled. But in cabbages, the third circled compound is also formed in substantial amounts. If you work in the risk assessment and registration of pesticides, you need to have a head for chemical structures. I really respect these people. They need to consider all of the different metabolites formed in different situations, how persistent they are, and how toxic they are compared to the parent molecule. But the question was, are metabolites important in the context of routine analytical chemistry? To the contract laboratory that's testing cornflakes for pesticide residues, river water for traces of pharmaceuticals, or whatever? The answer is an unequivocal yes. All of these types of compounds are unlikely to be present in food or in the environment just as the parent molecule. They are all biologically active and will all form metabolites. But it is one thing appreciating that you need to consider metabolites, quite another to decide exactly which ones you need to include, just as importantly, which ones not to include, and how you're going to approach measuring them. Clearly, you can't consider them all. The metabolites that you need to consider depends on the purpose of the analysis. This choice might be very different if you're measuring legal compliance with the maximum residue limit, or if you're trying to investigate where an illegal residue came from. And different again if you're trying to estimate the toxicological risk to consumers or wildlife. I'll run through an example of what I mean by each of the four. The easiest and most clear-cut one is legal compliance. Pesticides and veterinary drugs have maximum residue limits set in food. And when these were set, the legislators have already considered all of the metabolite data from the registration studies, which metabolites are toxic and which are persistent. The MRL is defined as the sum of all the metabolites they considered significant. So, for example, the MRL for malathion, an insecticide, is defined as the sum of malathion itself plus its maloxon metabolite, as you can see from this screenshot from the UK Health and Safety Executives database. So if you're testing a food for compliance with the malathion MRL, you need to measure malathion, maloxon, but no other metabolites. But you have to be careful. There are a few isolated examples where the residue definition might be different for different foods, or even for the same food under different legislation. This is not very common, but one example is abamectin, another insecticide. It's registered as a pesticide, but it's also registered as a veterinary medicine to treat ticks and lice on cattle or sheep. If it was used as a veterinary drug, the bottom box, then the MRL in sheep fat is just the parent molecule, called abamectin B1A. But if it was used as a pesticide, then the MRL in sheep fat is the sum of a list of other structurally related compounds. As an analyst, if you're testing samples for regulatory enforcement, then it's obvious which legislation you should be comparing against. But if you've just been sent in a sample by a supermarket, say, then how are you to know whether any residue you find is from pesticide or veterinary drug treatment? Which definition should you choose? When you're not dealing with a registered chemical, but are trying to check for illegal use, then it can be a bit more difficult. If you were acting as a policeman, looking for evidence, then you want to be looking for the metabolite that is the clearest signpost, the one that's easiest to detect for the longest period of time. you're not particularly concerned about the most toxic metabolites. But if there's not been a registration study, then the metabolites might not be particularly well characterized. You might have to hunt around in the scientific literature a bit to find the most suitable one. This example is one that's now well known, but was a surprise when it was discovered in the 1990s. Furazolidone is an example of a nitrofuran antibiotic, which had been banned in food animals for a number of years. In the early years of the ban, monitoring schemes did not detect any problems, but they were testing for the parent drug. Nitrofuran parents are metabolized within a matter of hours, so unless you happen to test the chicken on the same day it was treated, you'll never detect it. Once the protein-bound metabolites were characterized and methods developed to test for them, then, hey presto, it was found that nitrofurans were still being used extensively in food production in Southeast Asia and Brazil, amongst other places. The third reason you might want to consider metabolites is in diagnosing the source of a contaminant, to investigate whether it has an innocent explanation or if it's some kind of illegal use. This is easiest to illustrate with an example, as there are lots of different ways this might work. I mentioned pyrimecarb before. The MRL definition of pyrimecarb is only the parent molecule, so ordinarily you would not test for metabolites. But say you found a residue on a product that was labelled as organically produced. A frequent, quite plausible, explanation for trace residues on organic crops is that it was accidental cross-contamination. But if you tested for the metabolites and found them in substantial quantities, then this would be a strong indication that the pyrimecarb arose from when the crop was still growing, but it was applied in the field, either to the crop itself or to something in close proximity. The farmer would have some explaining to do. Finally, you might be conducting studies on toxicological exposure, either to consumers or to wildlife. Usually, metabolites are less toxic than the parent. The whole point of them, after all, is for an organism to try and clear an unwanted chemical from its system. But occasionally, metabolites can be more potent than the parent, or have a different and more serious mode of toxicity. In this case, you would definitely need to measure them. A good example of this is the difference between carbadox regulation in the EU and the US. Carbadox is banned in the European Union, so analysts are purely interested in detecting its use. They test for the major metabolite that persists for the longest amount of time, QCA. But in the US, carbadox is still used with its toxicology under active review. There is a very minor metabolite, desoxycarbadox, that contributes disproportionately to the toxicity of residues. So US analysts will often be interested in this as part of their testing. So the next question. Now you've decided what metabolites you want to consider, how do you actually measure them? At first sight, it might seem quite straightforward. Back to pyrimecarb. This is a page from the Dr. Ehrensdorfer catalogue, so that you can see that most metabolites people would be interested in are available as reference standards. But, like most things in analytical chemistry, it's not quite that simple. I'll run through 5 different approaches to measuring metabolites. Each of them is appropriate for different situations and will give a slightly different interpretation for the result. The simplest approach is just to treat each metabolite as an individual analyte, by a reference standard, calibrate, quantify it, then add all the results together. But, whilst I'm sure the reference material sales teams would love this approach, it's often not practical for laboratories. If you think of a typical pesticide residue screen, laboratories are testing for 300 to 400 pesticides. If you add in all the metabolites in the MRL definition, this becomes more like 600 to 700 analytes. As well as the cost of the standards, this would add a massive overhead in terms of method validation and calculation of routine quality control and calibration data. But as well as the expense, there are technical difficulties. Metabolites tend to be awkward to analyze. By their very nature, they're chemically different to their parent. They're much more water-soluble, or much more fat-soluble, or bound onto proteins. Now, a limiting factor can be the technical ability to incorporate such different natured chemicals in a single multi-residue test method. So the approach that's pretty much universal in pesticide residue analysis, and also common in veterinary residue analysis, is to use what are called marker residues. One molecule, or a limited selection of molecules, are chosen but will always account for the bulk of a residue pattern if a pesticide was present. If these are detected above a certain threshold, then the sample is retested. This time, rather than the 300 pesticide screen, the sample is only tested for the full metabolite definition of the one pesticide detected. This is an example. You can see that for the pesticide phorate, the laboratory here has included phorate sulfone as their marker residue. Phorate sulfoxide is there too, but this is more relevant to water samples. I'm guessing that they will use the same list for both food and water. It will be highly unusual for them to detect phorate sulfone above their screening threshold, so most samples will just be reported as negative. But if they did detect it, then they would need to retest for the 4 substances included in the MRL definition: phorate parent, plus its sulfone, oxon, and oxon sulfone metabolite. Now, I definitely don't want you to try and memorize this slide. The point is, again, that metabolite patterns can be extremely complex. But this complexity itself can be used as a testing tool. If you try to spot evidence of a steroid abuse in sports anti-doping, or in fact in raising beef cattle, by testing the concentration of a particular metabolite, then you'd generally be onto a loser. This is because the same metabolites are also present from the natural presence of hormones in the animal, and the variation in concentration from animal to animal, or even from day to day, is greater than the elevation that you would see following steroid administration. But it might be that the steroid gives a more subtle change in the overall pattern that's not apparent just from measuring a few metabolites. In principle, you can use statistical pattern recognition software to spot patterns and clusters. This involves measuring and quantifying a whole range of different metabolites and markers, even those where you don't really understand what their function is in the metabolic route. You can then use multi-component analysis to plot these in multi-dimensional space. I thought it a bit easier today to visualize in 2 dimensions. So in this example, you test a reference set of racehorses that you knew were untreated, then a reference set that you knew were treated with a steroid. Hopefully, the results would fall into 2 distinct clusters. So when you then came to your sample, if it did not fall within the untreated cluster, then you'd have some grounds for suspicion. It could have been treated with steroids. Although, unfortunately, this is rarely definitive. Unless you've thought of every possible innocent variation within your reference set, then there might be any number of other reasons why your sample is different. It might have a different feeding regime. It might be stressed. It might be happy. But it is suspicious. Metabolomics is not a routine technique. It's expensive, it needs carefully constructed databases and high specification equipment. So it tends to be reserved for research applications, for testing in elite sport, or for particularly high-profile investigations. The next measurement approach is to convert all similar metabolites to a common product and then measure that. This approach is written into some MRL definitions because it was the approach used to generate the safety data that was submitted for products registration. Ceftiofur here is an example. The MRL is defined as the sum of any metabolites that contain the betalactam structure, circled in green, expressed as a common product, desfuroylceftiofur. So any regulatory testing has to use the same analytical approach. Now this is the Ceftiofur metabolic pathway, with all routes going through desfuroylceftiofur. So in the lab, you force all of the different metabolites in your sample to convert back to desfuroylceftiofur. Unfortunately, there's an added complication here. Desfuroylceftiofur is not that stable, hence it tends to metabolize to other things. So most methods will convert it quickly to desfuroylceftiofur, which is much more stable, and measure that. Most laboratories will derivatize their desfuroylceftiofur calibration standards in situ from a Ceftiofur reference standard, so that the calibration includes the whole conversion process. The final approach I'm going to describe is to measure a particular part of a molecule that's common to multiple metabolites. This is the basis of immunoassay test methods, like ELISA kits. If an antibody is raised to a molecule that you want to measure, like fluphenicol here, an antibiotic, then it works by latching onto a part of a molecule. But this means that it will also latch onto similar molecules, like metabolites, such as fluphenicol alcohol or fluphenicol-amine. I won't run through them all, but you get the idea. You don't even need to know what the metabolites are. As long as they have a similar functional group, then the antibody might cross-react with them to a certain degree, and they'll be detected and included in the measurement. So with this approach, you have no option but to measure the sum total. So in conclusion then, it is vital to consider metabolites in trace organic residue analysis. It's an integral part. But it's very important to understand for each individual application which metabolites should be included and why. And as importantly, which should not. Finally, there are a number of approaches to considering metabolites as part of a measurement. Not all of them need reference standards for each and every metabolite on each and every occasion, but they all need to be checked and verified at some stage with reference standards. There are many parent compounds and their most common metabolites in our Dr. Ehrenstorfer range, available to purchase from LGC. Thank you.